Funding for this podcast is provided in part by the IPM Network through the USDA National Institute of Food and Agriculture and by various commodity groups through checkoff dollars, including the United Soybean Board, National Corn Growers Association, and Cotton Incorporated.
The I See Dead Plants podcast shares the stories of people and plants, pests and pathogens, and the conflicts among them. Join us as we speak to the folks who attempt to solve complex problems with plants through pest management research. I See Dead Plants is an extension-based podcast created by the Crop Protection Network and the North Central Integrated Pest Management Center and hosted by Ed Zaworski.
Welcome back to the I See Dead Plants podcast. As always, I'm your host, Ed Zaworski. Today, I am coming back to you after a month-long hiatus here, so welcome back to the folks listening. And we're going to be talking about citrus greening today, and I'm joined by Dr. Lauren Diepenbrock from the University of Florida. How you doing, Lauren?
Doing well. Yourself?
I'm doing fantastic. I'm glad to be back recording. So, as always, I'm just going to give a little bit of background information on Lauren, just so you guys get to know her a little bit, ask her a few questions. Got her bachelor's in biology at the University of Missouri-Columbia, master's in science education at Syracuse University, then a master's in ecology and evolutionary biology at Florida State University, followed by a PhD in insect and microbial sciences at the University of Missouri in Columbia. She's currently an associate professor of citrus entomology and an extension specialist at the University of Florida. Did I get all that right, Lauren?
Yeah, quite a bit.
Well, I was telling Lauren before we recorded, I love backgrounds like this because I love hearing the story of how people got to where they are. So that's my first question for you, Lauren, is how did you end up in the field of entomology?
I was not a direct path. When I went to college, I was the first in my family to be really into science. And so, of course, if you're going into science, you're going into medicine. So I went in as the pre-med track, realized, oh, heck no, that is not for me. Started working in a laboratory doing molecular biology work with C. elegans, particularly looking at some of the aging aspects. I had a really fantastic experience working there. Decided to apply for, at the time, a PhD in biomedical sciences. I had a really lovely offer at Upstate Medical University in Syracuse. Moved up there and with 6 months realized I hadn't seen the sun in 6 months. And so I was miserable. I am originally a Florida grown child, wound up in Missouri just because of parental job changes. And I dropped out of that PhD program because I was pretty miserable not seeing the sun. Realized, hey, I love science in high school. How about I get a science ed master's? I'm just going to go teach high school. That sounds fun. All right, so lucked out, got into the science education program at Syracuse University, which was a fantastic program. Wound up getting my master's there and then moved back to Missouri and taught high school science, biology, freshman chemistry, and AP biology for 2.5 years. My brain was not engaged in what I was doing. I very much enjoyed being a part of these students' development, but I realized it wasn't enough for me. Like, I needed more in my life. And so I started applying to programs thinking, you know what, I took this awesome ecology program or ecology course in my science ed master's. What about ecology? Like, could I get a degree in ecology and what could I do with that? So I applied to all these ecology programs with my interest because I had a background interest in medical sciences. And I was thinking, oh, it can be kind of cool to look at disease ecology, and then I wound up getting an email from a professor at Florida State, Walter Schinkel, who is now retired. And he said, hey, we've got this really interesting disease in fire ants. Do you want to come do your master's here? All right, cool. Tallahassee's a great town. Florida State had a really awesome program. I moved there for 2 years, did my master's in ecology and evolutionary biology. It turns out the pathogen had pretty well worked its way out of the system locally by the time I started. Huh. So I got there and I was like, oh, crud, what am I going to work on? And I was in a really lucky position that I taught through my whole master's, so it wasn't attached to a grant. And I helped a postdoc in the field collect some ants for collaborators in Canada. And I was like, man, these are the coolest ants. They were trapjaw ants. And if you know trapjaw ants, they have like massive mandibles and they snap shut on you. And like, not only do they snap shut on the front end and that hurts, but they have a wasp venom in their booty. So like they're snapping shut on you and they're jabbing you with their rear end and it's so painful. but they're so cool. So I asked all these questions, and finally he was like, nobody knows. Like, nobody knows this information. Why don't you do that for your master's? So I did my master's looking at natural history, just understanding the natural history of this organism because it hadn't been documented. So I worked with Odontomachus bernaeus, incredible group of trapjaw ants. They were so much fun. Realized I absolutely loved natural history, loved being out in the forest, in the fields. And I decided I wanted to do a PhD, but I wanted something a little more like a little more than what was there. I wanted actually a PhD in more entomology focus instead of evolution and ecology because I really didn't have the entomology background. And so I started looking around, reached out to a professor where I had done my undergrad, said, hey, do you have anybody interested in this specific topic? And I was really thinking about community ecology and conservation questions. And she, just as luck would have it, she hooked me up with a new professor. And so I was on the startup funds for Dr. Debbie Finke at the University of Missouri.
Okay.
And I came in and got to study coccinellids, or lady beetles. And I did lady beetle community ecology in tallgrass prairie systems and fescue. It was amazing. And I, during that time, met some growers because I had to find some fescue fields for my agricultural comparison. The extension folks helped me out. It was my first experience interacting with extension. They were amazing. And then I really enjoyed the growers that I interacted with, which kind of got me into this thinking about, hey, how can I do ecology and extension and like make this a job? And so after I finished my PhD, I was very, very fortunate to get an amazing postdoc at North Carolina State under Hannah Barak, who's now at Michigan State as the department chair. And I was working on spotted-winged Drosophila, which is an invasive pest of berry crops. And so I spent 4 years working in berry crops in North Carolina on this specific pest. It was incredible. I learned so much about pest management, how I could apply my ecology background into pest management, and just interacting with growers. And I
Yeah.
loved it. Like, there was nothing I didn't love about this job except for the pay rate of a postdoc. Um, so I applied for basically every position that had an extension role at that point in time because I was like, I really like this whole extension thing. This is awesome. Just as luck would have it, I interviewed down here early, I think it was on Valentine's Day of 2018.
Ooh.
The committee didn't realize it was Valentine's Day. So like I came down here, I had my bug Valentines to give out to the committee because we had the committee dinner the night before, so they got a good kick out of that. And I had a job offer within a week of my interview. It was just such an amazing experience coming here. So I never thought I'd be back in Florida. I mean, I don't know, maybe an hour and 15 minutes from the house I grew up in, and I have family within like 2 hours either direction. So it's kind of amazing to be back down here and to be working in an iconic cropping system despite the challenges that we have. Yeah, it's been a really interesting ride and nothing I ever would have expected in life.
I imagine that being an entomologist in Florida, there's probably an immense diversity in insects down there,
hmm
but I don't actually know. But
Oh, it's amazing. Like, if you like bugs, Florida is like the place to be in the US because there's just, not only do we have all of our endemics, and we have a huge difference from North Florida to South Florida as far as the soil types, moisture presence, plants that are here, habitats. Like, we have such a diversity of organisms between the plant, animal, fauna, animals, including insects, and snails and mites. It was just absolutely amazing system to work in.
I hear a ton about like reptiles in Florida
Yeah.
and, non-native reptiles
Oh, yeah.
specifically, right? Is
Oh,
it
yeah.
similar on the insect side? Or are there just tons of
There's quite a
exotic
few.
insects as well?
Yeah, there's quite a few. So since I've been here, we have worked with 2 organisms that are basically new to the region, and then one that has been in the region but was new to citrus. And so fairly, you could pretty well say 3 new to citrus in Florida pests.
Hmm.
And I've only been here 7 years, something like that, going on 7 years in May, I think.
That's probably a much, if you took the average of how many new introduced pests that the average new faculty member has to deal with in their first 5 to 10 years, that's probably pretty high, right?
Oh yeah, the first one was super nasty, and we found it. The growers found it the fall of my first year. And then they were just like, seriously, new entomologist, new bug. Did you do this? I was like, no. Actually, they called me out thinking it was cottony cushion scale, and being an entomologist who's interested in biological control, I was like, sweet, I've never seen cottony cushion scale. They weren't happy with my excitement until I got there and I realized that ain't cottony cushion scale. That's something else entirely.
Ah,
So I had to work with the state taxonomists to figure out what we had and then start developing some management research.
man. Sounds like a blast if you're an entomologist, of
I
course.
mean, it's frustrating and exciting and all the things in between.
Right. Before we got to the main subject today, I wanted to ask you one more, Lauren, and it's I don't know. I mean, since you had a background in science education, I was just kind of curious how I mean, I'm assuming that has helped you as an extension person, but I don't know what different
I think it
perspective has it
has.
given you maybe?
I think it really helps. I tend to approach my presentations, whether they be academic or extension, a bit differently than a lot of my colleagues. And there are a lot of things that, because of my education training, I think, yeah, everybody knows this, but they don't.
Ah, yeah,
And so
yeah.
sometimes when we're putting together extension programming, I have to remind people, hey, the average adult has a 20-minute attention span. That's all they got. So don't give a 40-minute extension talk, give a 20-minute talk, break it up with things. And so we do some different approaches. There's a core team of extension specialists here: entomology, plant pathology, horticulture, and until recently, economics. So that position hopefully will be filled at some point because he just left.
Hmm.
But we do a lot of the planning and a lot of the guidance on how to do the extension. And we are very lucky we have an extension manager and support team here, because we're not, a lot of systems you have winter, right? You have a down period. We don't have that in Florida. We are 12 months out of the year busy, 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 busy in the
Yeah.
field. So, it's a lot of times you have that period in the winter where you're like, okay, I could do all my extension programming, I could do all my workshops, growers are able to attend.
Hmm.
Doesn't work in Florida.
But yeah, I just saw that and I was like, man, what a great combination because we I talk with my colleagues all the time about the things that you thought that you were going to do with your degree and the things that you didn't realize that you were going to do with your degree and how they're vastly different. And I don't know, I think a lot of us don't have a good grasp, like you said, on how to educate and what are the best practices and that kind of stuff. I've taken some courses myself and learned a few things, but certainly not a science education degree. So it's got to be a
Yeah,
good
it's super valuable. I mean, I kind of wish there were more opportunities to collaborate within that area of extension and in pure education training, because I think we could grow a lot.
There should be. You got any I mean, maybe it's the length of somebody's presentation that they're gonna give. You got any tips for extension education folks across the nation?
Make it more interactive. Just make it more interactive. The more people see, touch, feel, use all the senses, use hearing, use sight, use touch, use polls, anything that keeps them interacting with the material and with you is really good for engaging all the senses. The more senses you engage, the more likely people are to remember things.
There you go, folks. That's actually something that I strive to do based on a class that I took here, so it's good to know that you agree.
Yeah, it's so important, and it's harder to do with certain topics like pathology. I mean, it's an ooey gooey thing on a plate. So, like, that's a hard one. I have 3D printed bugs, and so, like, next week I'll be in Georgia. Most things are fairly dormant right now, so I can't bring live specimens across state lines. That's a big fat no-no. So I've got 3D printed bugs so people can see. I
Yeah.
mean,
I've got a 3D printer running on the other side of a wall from me right now. I think it's actually printing some 3D bug keychains, some stink bug
nice.
keychains right now. So
Very nice.
we're working on pathogens, but it's a little bit harder to do. Print a big
Great.
spore. All right. Well, let's jump into the main event here. Let's talk citrus greening. And before I get to the vector pathogen system itself, I kind of wanted to ask you because I don't know anything about the citrus industry, and I'm sure that a lot of my listeners also don't. Can you kind of give an overview, Lauren, of like what goes into citrus? Where is it at? What types of citrus are we talking here? Like, what do you got going on down there?
Yeah, sure. So I'll give you a little historical perspective and then current day. So I'm only going to focus on Florida. And I will
Yeah.
mention where we've expanded. So we pretty much grow citrus north to south. There's different varieties that you can grow in different regions. From Satsuma up north, which is really a lovely, delicious fruit. They should be harvesting pretty soon. Maybe I'll get some on there next week. And then we grow sweet orange, grapefruit, pomelo, some lemons and limes down south. A more recent variety is finger limes, which
Okay.
is an Australian species, but we have a breeder here at the station who has been developing some really cool finger lime varieties. If you have never tried a finger lime, like
Mm-mm.
spend the money to try it at a store. They're stupid expensive. But when you pop them open, it's like little balls of caviar and it's like the
Huh.
most sour lime candy you've ever had in your life.
Oh,
It's so
interesting.
good. If you like sour, totally try it. They're using it as a garnish in a lot of places, like fancy restaurants for drinks and foods. I had some at a restaurant in Australia where it was like a garnish on, I forget what it was, like clams or something. No, not clams because I don't like clams. It was something else. But yeah, it's pretty cool. We do the whole range of, you name it, citrus. We'll pretty much grow. We don't have blood oranges, which is quite unfortunate. I very much like those, but that's more of a Texas, I think,
Yeah.
South
What was that first
California.
one, Lauren? What you said Satsuma?
Satsuma, they're these
I'm not familiar at all,
oh,
so
man, they're so good. I don't know if they ship them up into the Midwest, but they grow them in North Florida and in South Georgia, and they're these fat, juicy citrus, and they're yummy. I mean, it's like candy. It's so good.
is it like anything else, or is its own thing?
It's comparable to a sweet orange, which would be most of
Okay.
what you'd see in the grocery store, but my experience has been that the flavor is just a little different, and I can't explain how it's different, but it's really yummy. I highly
I'll have to
recommend.
keep my eye out. I don't know if we ever get them, at least in my area, but I'll keep my eye out, and
Yeah, and
certainly if I'm in Florida, I'll keep my eye out.
Are you interested in reading the latest integrated pest management updates, such as current pest concerns, management tips, research updates, available IPM resources, upcoming events, and job announcements? Make sure you subscribe to the monthly newsletter, The Central Issue, published by the North Central IPM Center.
yeah, and so when we produce, most of the citrus production is done on very sandy soil, because that's what we are. We sit on sandstone down here. And so a lot of the citrus, I joke that it's hydroponically grown because it's basically trees in sand. And we have added some cover crops to add nutrient profiles in some areas, which has been really cool research that my colleagues have been doing.
Mm.
Um, to see if we can do better with our nutrient retention. So yeah, we can grow up from north to south. We grow up mostly in sand. There's a rootstock and scion combination. So the rootstock is the below ground part, and that's going to be picked based on your local areas. So basically, we have different soil types, and those rootstocks hit those different soil type needs. Some of them will be a little more phytophthora resistant, which is a soil pathogen. And so there's been a lot of work done on rootstocks. We have guides for the whole state, and then they're kind of painful to read, to be completely honest. But if you work with an extension agent or one of the breeders, they can really help you narrow down your best choices. And then they graft scions onto the top of it. So scion is the part that we eat, and so
Gotcha.
that is going to be the variety of whatever is coming out and going to the market. And so yeah, we have a whole bunch of different scions, and generally those are going to be picked based on where you are, what you want to grow. Some of them are less favorable to certain insects. We haven't come up with a fully resistant scion for citrus greening yet. That is the dream. That is the dream. And so, yeah, citrus used to be, when I was a child, I'm not even going to give you the years, most of the state was covered in citrus. I mean, you could smell the bloom. I grew up on the East
Mm,
Coast. My grandparents were on the West Coast. You could smell it driving across the state.
mm.
Now we've lost, I want to say it's like 90% of our acreage.
Oh, wow.
It's really, really high, and it's all majority because of citrus greening,
Oh,
and the costs associated with it. And so we've lost a ton of acreage. There's a lot of jobs that have been lost. There's some really nice economics research that has actually gone into the financial impacts of losing so much of the industry. We've lost packing houses. So it's kind of a depressing time, but at the same time, there's hope. We have some tolerant genes that look like they make the trees tolerant. They look promising. They're in the field right now. We'll know in a few years what these trees really can do.
Hmm.
We have a couple tools that are being implemented, and we could talk about those later on too. And so there's some promise. There's always hope. Growers are a unique group. They're very ingenious in how they think about things, and sometimes their workaround is absolutely brilliant, and we would never have come up with it. So that's the cool part of being an extension specialist is I talk to the growers all the time, and
Yeah.
sometimes they have just the most amazing ideas, but they're not sure and they don't know how to test it. So I've had some really cool research projects developed in partnership with my growers to try to figure out how can we still make this work. Um, and so yeah, we have a lot of challenges, and it's a high input system right now because of the pathogen, and yeah, it's not fun.
I've heard that so many times, like, about taking a backseat kind of as a researcher or as whoever and listening to the growers and working with them in that sense. So that's good to hear. Lauren, like just in general, with a citrus tree, I'm just curious, how long does it take to get one to the point where it's producing fruit and how long are they like viable, I
Okay, so
guess,
that's going to be
depending.
somewhat variety specific, but also how you're growing it.
Sure.
So you're giving me a great opportunity to talk about some of our tools here.
Oh, good, good.
Historically, you'd put a tree in the ground and you'd start getting fruit maybe about 5 years at least before you start have like good fruit. You'll have crappy fruit before that, and you basically toss it because you know it's not worth anything. But this is one of those ingenious grower ideas where they said, hey, can we put bags on these trees and keep the pathogen out? And so they came up, these two growers that they come up with all sorts of cool things. These guys are amazing. They came up with this thing and they're calling them individual protective covers, IPC for short. And
Hmm.
essentially what they are is exclusion mesh. And it's small enough to keep the Asian citrus psyllid out, which is the vector for the pathogen. And so if you grow a tree under an IPC, you will start getting fruit, viable fruit, in about 3 years.
Okay.
Yeah. And if you grow a tree, we also have what are called cups. So that's citrus under protected screens. And those are large greenhouses. The trees are there's some with trees in pots, most of them are trees in the ground. And it, again, exclusion mesh. And there's other challenges in these systems. But without all the other challenges, those trees have fruit in 3 years and you can start making money. You come out of the red in about 5 years.
Gotcha. So let me make sure I
Yeah.
understand. So prior to citrus greening, you could get viable fruit that was saleable before, well, at about 3 years versus about 5 years. But now with the inception of citrus greening, it's pushing that back. Is that?
Uh, no, not exactly.
Oh,
So I
okay.
probably didn't explain it right. So open grow citrus, say I was just taking a young seedling and putting it in the field. It's already had its time, right? So like the scion's grown on the rootstock in the nursery for a while.
Okay.
And then in the field, they'd be producing a decent amount of fruit, decent quality fruit in about 5 years. This is pre-greening.
Oh, pre-greening. Okay.
Yeah. And even with that, so the challenge with greening is that it actually affects the vascular system of the plant. And so things don't move right. And so those trees often don't come into production if they're not protected when they're young.
Uh,
And so that's where we're like, if we have those exclusion mesh bags or the cup or the screen houses, the trees can actually grow. And it's kind of cool because some of them can self and they don't need pollinators. And so
uh,
we actually can get some fruit. We had a trial that was planted, and this was before we knew that. And we started getting really humongous fruit under these IPCs in the third year of that trial. And we're like, what is this? So, yeah, it's something we've learned, and it's really a pain in the butt to pick from under an IPC. So, there's specific instances you use that.
mm,
And then the growers basically you'll leave an IPC on for well, we're still figuring out the exact optimal timing, but a year to 2 years, and then it basically outgrows the bag.
mm,
And then you have to open it up, and then you have to start managing for pests.
gotcha.
And if you manage for well, I mean, you should be managing for other pests inside the IPCs, but like you'll be managing for the one that moves the pathogen around.
Gotcha.
It's a whole like complex set of things, and we don't have recommendations right now for how long to keep the IPCs on, and like basically what you do after you take it off exactly. So we do have a grant to look at this, and our field will be planted as soon as we are past the last freeze date for this year.
Gotcha.
Um, that's a huge project involving I don't even know how many labs, but we have it here and then also in South Florida. So stay tuned.
Yeah,
We'll have
yeah,
some
yeah.
guidance in a couple years. The growers have been kind of working on it without, before us, because we can't really do the work till we have grant money in hand because we have to pay people to do the work.
Sure. So, generally speaking, you're getting some fruit after about 5 years. How long does a particular and I yeah, it probably depends on the variety, like you said, but
Yeah.
on average, like, how long is a tree producing fruit before it's taken out of the system?
So if greening is not in your system, they can live for decades and be
Uh,
productive.
okay.
But with greening in the system, not very long.
Gotcha. Gotcha. Gotcha.
Yeah, so it's a huge change. I mean, historically, growers used to be able to plant their trees, and they'd only have to do minimal pest management, and they would have these amazing yields, amazing profits. And now it's like there's so much more management action that has to happen, and there's just so much variability in what you're going to get out of that management.
Sure. So, I mean, obviously, we're talking citrus greening here, and that's the heavy hitter. But what are some of the other challenges that citrus runs or citrus growers run into with regards to pests and pathogens?
Um, so I'm not a pathologist, but I do
Sure.
know a few things there. I know that melanose can be a problem. Greasy spot can be really problematic. More recently, Dr. Dudeney's been talking also about greasy green, which is related to greasy spot. And don't ask me the difference because I can't tell you. Uh, Phytophthora, which is a root pathogen, can be hugely problematic in certain regions. Some areas are more prone to it. And also, we have a root weevil. So if you have the root weevil in your system, it chews on the roots, it opens holes, the Phytophthora gets in easier. And so there's various factors. Also some nematodes in the system, which can also make holes for Phytophthora to get in. So we have things. We have scales, we have mites, we have mealybugs, we have stink bugs, leaf-footed bugs, you name it, man. We've got all sorts of good stuff down here. And more recently, we have an invasive snail that has been causing a lot of problems.
Oh, good.
I've been learning a lot about snail biology because they're not insects
Mm.
and they do life very differently.
I got one more for you before I
Yeah.
want to get into like the fine details of citrus
Yeah.
screening itself, but just in general, like obviously you start talking about some of these what was it IPCs, the
Yep.
individual protective covers. I mean, beyond that, like what's the typical management that a citrus grower is I mean, it sounds like there's a lot of things that are challenging these
Yeah.
citrus trees, but I mean, just kind of at a very basic level, like what are they doing? Or is there a lot of fungicide, insecticide applications?
Yeah,
What's
so
the
it
typical
depends.
management?
Oh, I forgot canker's another one of the pathogens they have to deal with. So, yes, they do have to treat fairly regularly. A lot of the growers are kind of what's left of Florida. We don't have a lot of small producers anymore. Basically, the large producers are
Mm,
left for the most part. So a lot of them have to do by calendar because they have to move equipment between fields. That's never the ideal,
right,
but it's just a reality of running a business.
sure.
So for insects, um, again, historically, there wasn't a lot that they had to do. But once Asian citrus psyllid came on the scene, there was a lot of research going into how to manage this pest. And so they wound up spraying like 12 to 14 times a year for it. We don't do that anymore. We've learned more about the insect and its life cycle and where it's tied to. So we only do, depending on the market, most growers do around 6 treatments for Asian citrus psyllid.
Mm,
Just Asian citrus psyllid, 4 to 6, just depending on who the grower is, what the market is. Um, if you're so Central Florida, where I am, is mostly juice producers, so we could have some more
uh,
blemishes. And then on the coast is where you have more of the fresh fruit, so they have to spray a lot more often because they have to have perfect fruit.
mhm,
So there's a balance there too of like who is your market, and that's always something you have to think about when you're a producer. And so yeah, we manage for several different things. Rust mites are always problematic, and we know the approximate timing of those rust mites based on the seasonality and the weather. They have to do canker treatments at certain times of year where they're putting out copper like every couple weeks.
mm,
I don't remember the exact regimen. If you wind up talking to Megan, she could tell you. Also, everything's in the citrus production guide for all of our recommendations. So we have a book that we edit every year with all the recommendations. Fertilization's critical,
mm.
especially with these sick trees. They've been doing a lot of work with nutrients. It's really incredible work, and it's kind of one of those things where I'm like, whoa, I never thought about that, but duh, it makes sense. So there's a lot of different things going on at any given time. So like as an entomologist, I focus on bugs, but I think it's really important to take a step back and realize that no person is just managing the bugs. And so, like one of my first really big projects here, it was amazing. I got a project that was whole grove management. And
Okay.
so it was horticulture, it was pathology, it was a fertilization and irrigation person. And it was this crazy huge project that we started right before the station went on pause for COVID. Literally, we're planting right before we were told to go home.
Oh, no.
So it was really complex in more ways than we had planned, but it was a truly incredible experience. The data have been so useful, and we learned so much.
Hmm.
We're still publishing data out of that experiment. It's been over for a while.
I bet that the handbook that has all of the recommendations in it is very thick.
It's dense. It is dense. We're in the middle of editing it for next year, so we make edits every single year in any updates. And yeah, that's I'm the lead for that, so that's one of my biggies. But I have two other co-editors who are amazing, and like the person who handles horticulture has to be a saint because there's so many people in the horticulture field, and they don't all agree.
Oh, wow.
So
Yeah.
she's amazing, and I'm glad it's her, not me.
Okay, well, at this point, I might come up with some more questions as we
That's
go
fine.
along. I usually do is with regards to citrus, and I'll try to pepper them in if I think of them. But citrus greening. So, for those who don't know, what is citrus greening and what is involved in it?
Okay, so citrus greening is caused by a pathogen called Candidatus. Well, in Florida, anyways, we have Candidatus liberibacter asiaticus. There is also Candidatus liberibacter americanus and Candidatus liberibacter africanus. But all we have here that we know of is asiaticus. And so that particular pathogen, it's a bacterial pathogen, and it is transmitted by the Asian citrus psyllid, which is a piercing sucking insect. Now, there are other piercing sucking insects that can transmit, but this is the primary vector for it in the North America. If you were in Africa, a lot of it would be Trios eritreae and doing africanus. So there's different primary vectors depending on your pathogen and your region in the world. But again, we call it CLAS, so Candidatus liberibacter asiaticus, CLAS. Asian citrus psyllid, ACP. Those are the acronyms we use. And then citrus greening essentially is caused by the output of this pathogen. And so the pathogen, think about like sticky bacterial cells, right? So they have sticky bacterial cells. They're building up in the phloem of your plant. And I like to use a human example here. Now, it's not an exact transfer, but I think about cholesterol and arteries and how cholesterol clogs your arteries.
Yeah.
So think about these little bacterial cells, kind of like cholesterol molecules, like clogging the arteries. And so they're clogging the phloem of the plant. And with that, you're not moving nutrients through a plant correctly. And so you wind up not getting enough sugars put out to the fruits and leaves. Eventually, so you start seeing the pathogen above ground in leaf reduction, leaf distortions, leaf discoloration, corky veins. But you're actually seeing the later effects of it because the first effects are below ground. We actually start seeing impacts on the roots first. And so it eventually causes depletion of the roots because there's basically nothing feeding the roots. And so then it messes up the whole movement of like root to shoot, shoot to root that should be happening. And one of the wild things about this pathogen is they actually showed that like you can follow the pathogen seasonally. So like in the winter, more of it gets shunted down to the roots. You know how you store like sugars in your roots?
Yeah.
So you may not get a high titer from the leaves at that time of year because you're not moving nutrients up into the leaves. But if you sample the roots, you'll get higher titers of the pathogen in the roots. And then as soon as we get our spring flush, it goes right back to the leaves. So it's
Interesting.
really wild. And the research has been really cool on the pathology side of it. On the insect side of it, the psyllids feed on sick plants. And then they acquire the pathogen, and then when they feed on another plant, they essentially spit the pathogen into the phloem of the other plant. And they are phloem feeders, and so you can actually look at their feeding interactions using electrical penetration graphs, and you can see when they actually egest or spit, and in that spit is where the pathogen is. And so that's how it gets moved around. The juveniles are much more efficient at acquiring the pathogen than the adults. And so how this kind of works, if you think about a bigger system, is the adults are attracted to the flush to mate and lay eggs.
Okay.
So they go to the flush. It's the easiest tissues to feed on because they're really soft and tender. So they lay their eggs there on the flush, and we generally scout what we call finger flush, which is the teeny tiny little bitty flushes. So those are the little bitty leaves, things that will become leaves for people who don't know what flush means.
Yes.
Yeah. So I always tell my students to be really careful about that, and I just made that big mistake. So
No,
um,
you caught it.
yeah, I caught it in time. So anyway, so they lay their eggs there, and then when the eggs hatch, the juveniles, which are nymphs, feed on that flush. And that's the only material the juveniles can really feed on because their mouth parts are not like fully sclerotized like the adults are. And so they feed on that flush where all the pathogens getting shunted to. So they acquire it really readily.
Yeah.
And then it stays in their body as they molt through their life stages, and then they become adults. And then when they're adults, they're mobile, they fly around, and they can now infect new plants. So it's very tied to flush as far as the psyllid life cycle, which, opens up this doorway. This is part of why we could reduce some of our spraying, is that we were able to target flush periods. However, there is a challenge here. If you are messing the nutrient flow of a tree, you are going to mess up the signals of when it flushes. And so now, instead of having 2, maybe 3 distinct flush periods in our system, we have all this off-time flush. So instead of like, okay, I should have a big push of young tissues in March, I'll get a lot, but then I'll get more in April, and I'll get May, and I'll get June. So instead of like these major periods, we'd be like, cool, we could spray these 3 chemistries in these time periods and know we're killing the juveniles. Now I have, unfortunately, the appropriate habitat resources, whatnot, throughout most of the year. And so they're able to persist and keep increasing their population throughout a lot of the production season.
So hang on, what changed to go from, because you said at some point there was one flush and now there's multiple flushes, and is that
So,
because of nutrient inputs, you said?
yeah, so we used to have 2 to 3 really heavy flush periods,
Okay.
and you could, in theory, time your chemical management to those flush periods and kill your juveniles. And that was amazing. It was a cool idea. But with the changes in the physiology of the plant from the pathogen, it
Oh,
then causes
I got you.
off-time flush production. And part of it is just doing nutrient movement. Part of it is the plant's like, what the heck's going on here? And so there's been some really interesting research where they've tried to induce flushing. So put it into drought cycles and then irrigate at specific time points. And it
Yeah.
kind of works, but I'm not going to say I'm happy enough with the data that I would recommend it necessarily, because you're also stressing your trees. So you're then getting stress flushes, which are not, from my understanding, optimal. Could be wrong there. But we've done a couple different studies with several different groups to look at that question, and nothing's clean. None of the data are particularly clean. And part of that, too, is that the adults can feed on any of the leaf tissues.
Gotcha.
They just only lay eggs on the flush.
Man.
Yeah, it's tricky. And then there was a graduate student about like 4 or 5 years ago who looked at alternative hosts for citrus. And
Yeah.
so they can reproduce on most things in Rutaceae, but the adults can also take drinks from other plants.
Hmm.
And so, we were so excited with this big freeze. We're like, woohoo, kill the psyllids. No, it doesn't work like that. A lot of them there's a certain proportion that can survive a freeze. And then they have other resources in the system that they can get some nutrition from and be ready to go as soon as the flush starts.
Oh, man.
Mm-hmm.
Really quick, Lauren, for someone who doesn't know psyllids,
Yeah.
what is a psyllid?
So a psyllid is a very small, piercing, sucking insect. Picture, like most people I think know kind of what an aphid looks like, right?
Mhm.
So it's around the size of an aphid, but with like longer wings. So most of the time you don't see winged aphids aside unless they're stressed out or it's the right time for them to migrate to a different host. And so these are, as an entomologist, I think they look nothing like a psyllid, or like an aphid. However, in reality, like physiologically, they had the whole piercing, sucking mouth part. Their body's about this is kind of an elongate form of that. So psyllids are kind of like fat balls of goo. These guys are a little more elongate, and they have these longer wings. But size-wise, yeah. And the nymphs often get mistaken for aphids, even though, again, I think they look nothing like them. But I can see where you might be confused, especially if you're not looking under a microscope.
Sure.
It's a blob and it's shooting out honeydew. Except that one of the really cool things with psyllids is that they don't have the like liquid honeydew that a lot of piercing-sucking insects do, they make a wax. And
Oh,
so you see this, like, waxy tendril coming out of their rear end, and so that's there's no better way to say it, man. It's awesome. It's so cool to see.
thanks for listening to part 1 of my interview with Dr. Lauren Diepenbrock talking about citrus greening. Make sure you tune in next week for part 2. Thanks for listening, folks.
I See Dead Plants bridges the gap between plant science research and the impact it has on our daily lives. It is brought to you by the Crop Protection Network and the North Central Integrated Pest Management Center, which are both partially supported by the USDA National Institute of Food and Agriculture. For more information on today's topic or other pest management issues, check out our show notes, cropprotectionnetwork.org or ncipmc.org. Remember, this information is for entertainment purposes only. The statements made in this podcast should be interpreted within the limited context of the particular topics being discussed. Contact your state extension program for local information on pest management. For the full non-discrimination statement or for accommodation inquiries, go to cropprotectionnetwork.org/about. Music written and recorded by fellow plant pathologist Sean Toporek. Thank you for listening to I See Dead Plants.